Great. Well, fantastic. Thank you so much for attending my talk today. I'm very excited about being here with my work to you, which I hope you find of interest. And today I will be talking about the application of multivariate genomic methods to study the joint genetic architecture of health-related traits and aging-related traits, with a special focus on cognition, dementia, and chronic conditions. So, to begin with, I would like to first provide a very brief introduction to some that will be appearing through my talk and that are of particular relevance for genomic research. The first one is the concept of heritability. A broad definition of heritability is the proportion of observed differences in trait among individuals that is due to genetic differences. It is important to note here that heritability is population and time specific. It is not a natural constant, and of course, it does not imply biological determinism. The most common form of genetic variation are single nucleotide polymorphisms, where a single nucleotide, the box of the DNA sequence, is different between individuals at a genetic position. SNPs capture the majority of known genetic variation among people, and they're easily measured as categorical variables reflecting different allele states, indicating the number of copies of a reference allele, usually a risk allele, if we're talking about diseases that a person has inherited from their parents. So genetic architecture. Genetic architecture refers to the joint distribution of effect sizes and allele frequencies of the causal variants associated with a trait of interest. Mendelian traits are influenced by one or a few genes with very strong effects, such that if the person carries this particular genetic variant, it is very likely that they manifest the phenotype. One example of this type of genetic architecture would be Huntington disease. On the other hand, we have complex traits, which have polygenic architecture. They're influenced by hundreds or thousands of genes with very small effect sizes. This is the case for most psychological traits and psychiatric disorders, but also for some common medical conditions like type 2 diabetes or some circulatory or musculoskeletal disease. So typically, the larger the effects of the genetic variants, the smaller samples sizes are required to identify these variants. It is easier to detect individuals at risk, and the biological mechanisms underlying are easier to understand. For complex traits, however, small effect sizes, genetic variants with small effect sizes require larger sample sizes. It is more difficult to identify individuals at risk. And of course, the biological mechanisms are also more complicated to uncover and to understand. So the conceptual framework for genome-wide association is statistically very simple. It is based on a simple linear regression model where we estimate the effect of a SNP or a genetic variant on a phenotype of interest. And in genome-wide association studies, a simple linear regression model is performed for each SNP, resulting in millions of association test statistics and issues related to an inflation of false positive rates. The standard approach to deal with these issues related to inflation of false positive rates is to perform a one-for-one correction based on the notion that there are approximately 1 million independent tests in the genome based on the covariance structure among SNPs in the genome, or linkage disequilibrium structure, LD structure. This correction results in the genome-wide significant threshold of 5 × 10⁻⁻⁻, typically seen in genome studies. So another key concept that is particularly relevant for my work is the concept of pleiotropy. Pleiotropy appears when a single gene influences multiple traits. There are different types of pleiotropy. So there's vertical pleiotropy, where a genetic variant affects one trait, which subsequently affects other traits downstream in a causal chain. And there is also horizontal pleiotropy, where a genetic variant directly affects multiple and sometimes seemingly unrelated traits. With increasing sample size in genome-wide association studies, the number of hits or genetic variants associated with polygenic traits has dramatically increased. These are the Manhattan plots for two highly polygenic for one medical trait, atrial fibrillation and educational attainment in the top. So Manhattan plots are the graphic representation of genome-wide association study results with chromosomes and base pair positions in the x-axis and the negative p-value, the negative logarithm of the p-value for the SNP trait association in the y-axis. From a simple visual inspection, it would be very hard to find or identify the overlapping hits or overlapping genetic variants for these two traits, right? Thousands of hits for both of these traits. But luckily, we have genetic correlations, which are basically a quantitative measure of the degree of genetic overlap between pairs of traits. So recent large-scale GWAS have revealed substantial genetic sharing among chronic diseases of aging, as highlighted in this heat map of genetic correlations among some of the most common and prevalent medical conditions in the old age. We can see that there is a genetic overlap even between diseases from different organ systems, like osteoarthritis, disease of the connective tissue, and type 2 diabetes. These findings are very exciting. However, they come with a series of limitations. First, they're mostly descriptive and a summary of the pattern of genetic overlap at the genome-wide level. They're based on a bivariate approach across pairs of traits. And of course, they provide little to no understanding of the underlying genetic architecture for this constellation of traits. So this limitation is what motivated my colleagues Andrew Grotzinger and Michel Nivar, and my mentor at the University of Texas Austin, Elliot Tucker Grubb, to develop genomic structural equation modeling, which is a very general framework that allows fitting structural equation models to genetic covariance matrices using GWAS summary statistics. So these structural equation models represent different genetic architectures for constellations of traits at a genetic level, and they're very useful to identify mechanistic insights underlying the genetic covariance structure among constellations of traits. This has one of the strengths of this approach is that it allows to integrate phenotypes from disjoint samples measured at different time points across the lifespan. So we could, for example, estimate the association between birth weight and late onset Alzheimer's disease without the need for raw genotype data. And just mentioned, this is implemented in an R package. It is publicly available. There's a very nice wiki website where you can actually find some tutorials to start working with this package. So now I'm going to provide a brief overview of how genomic SEM works without getting into too much detail on the technical aspects of the method itself. So genomic SEM follows a two-stage approach. In the first stage, we estimate the SNP-based heritabilities and co-heritabilities and the genetic correlations using GWAS summary statistics. This is using the multivariate extension of LD-score regression. So this is how a typical SumStats file looks like. We have as many rows as genetic variants, and then we have effect sizes for the SNP trait associations, standard errors, and p-values. So in the second stage, the user specifies a structural equation model, which is a multivariate system of regression and variances association involving the genetic components of the phenotypes with one another and with one or more general factors representing broad dimensions of genetic sharing among particular clusters of traits. So as I mentioned before, genomic SEM, these structural equation models reflect different genetic architectures and are very useful to reduce dimensionality, providing a lower dimensional representation of the original genetic correlation structure. So genomic SEM also allows to integrate individual variants in the model to identify SNPs or genomic loci with effects on broad dimensions of cross-trait liability versus genomic loci that are more specifically relevant to particular traits or diseases. This is done by comparing a common pathway model that assumes that the effect of the SNP on the individual traits is fully mediated through the genetic factors, latent factors, versus an independent pathways model where the SNP directly affects or predicts the genetic component of each of the individual traits. So from this model comparison, we derive a QSNP statistic, which is basically an index of heterogeneity that tells us to what extent our common pathway model is a good approximation to the SNP trait association. And it's useful to safeguard against false discoveries on these genetic factors. So now I would like to present some of my recent publications applying genomic SEM to the study of cognitive function and dementia. This first paper was published in 2021, I think, in Nature Human Behavior, and it is the first formal investigation of the multivariate genetic architecture of human cognition using molecular genetic data. So for this study, we used genome-wide data from UK Biobank for 7 cognitive tests covering diverse cognitive abilities and processes. And we investigated the pattern of genetic sharing among these 7 cognitive tests. So prior to the formal modeling in genomic SEM, prior to fitting the structural evaluation models, we conducted some descriptive analysis of the cognitive traits, genetic correlations based on LD score progression. And we found a substantial but imperfect overlap among these cognitive tests, which resembles to findings that are typically found in phenotypic research of cognitive function, with genetic correlations ranging from 0.13 to 0.87. So the first principal component derived from the eigen decomposition of this genetic correlation matrix explained 62% of the genetic variance among the cognitive tests, which suggests the presence of a general dimension of genetic sharing among these cognitive traits. So we then proceeded to with genomic SEM to formally model the genetic variance structure, and we applied genomic SEM to fit a single common factor model to the genetic covariance matrix among the 7 traits. The model specified the genetic component as represent of each of the individual tests, as represented by the circles in the bottom of the path diagram, to load in a general single common factor, which we termed genetic G, and representing the shared genetic variance component among the 7 traits. But we also, for each trait, we additionally estimated residual trait-specific genetic variance components. So these numbers here are factor loadings, which are basically regression weights that relate the growth factor, the latent factor, and the genetic component of each of the individual traits. Okay, so this table here reports the proportion of genetic variation explained by the shared and unique variance component for each cognitive trait and the respective absolute contribution, the SNP-based variability, as estimated from LD score regression using HapMap3 common variants. So the general factor of cognitive function, this genetic G factor, accounted for 58% of the genetic variance in the 7 cognitive traits. However, interestingly, what we found is that a proportion of genetic variation in each trait accounted for by the shared genetic component differs substantially across traits. So, for instance, for TRAIL-B, which is a test assessing executive function, which is a higher order cognitive ability, 95% of the genetic variance for these tests is accounted for by genetic G, by the shared genetic component. whereas only 5% is actually specific to that particular test. So in contrast, for RT, which is reaction time, is a measure of a more simple cognitive process. The percentage of variation of genetic variation accounted by the shared genetic component as represented by genetic G was only 9.5%, which indicates that the genetic architecture of that particular trait was very unique and not shared with the other 7 components. This has important implications for genomic research, as univariate analysis, like standard univariate GWAS, of some of these individual traits, like TRAILS-B, will largely reveal results relevant to this general factor of cognitive function rather than to specific abilities. Okay, the next paper that I would like to talk about was published last year in PLOS Genetics, and it focuses on the genetic architecture of Alzheimer's disease and some biases in the estimation of heritability when combining GWAS of case control GWAS, which we call direct GWAS, and proxy GWAS of family history of disease. So genome-wide association studies of proxy phenotypes using family history of disease, parental history of disease, can substantially boost power when combined with traditional case control GWAS. This has been particularly relevant in the context of Alzheimer's disease and other relatively rare diseases. So recent meta-analysis, high-profile GWAS meta-analysis combining GWAS with direct GWAS data on Alzheimer's disease have expanded the number of AD relevant risk loci far beyond the well-established APOE variant, which is the flagship of the genetic markers for Alzheimer's disease, to actually to 75 loci in total. However, what about heritability? Well, despite a twin-based heritability estimate of approximately 60%, these studies, this meta-analysis, indicate very low common variance in heritability after excluding the APOE region, with the most recently reported estimate of 2.5% coming from the largest GWAS on Alzheimer's disease. And this calls into question the continued discovery or understanding of the genetic architecture of disease. So conventional approaches for analyzing GWAS and GWAS data either use an inverse variance weighted meta-analysis of regression coefficients, using weights as the inverse of the variance of the effect size estimates, or meta-analysis of C statistics with weights equal to the square root of the study-specific sample size. So for the inverse variance weighted approach, the standard correction factor for GWAS of the phenotype of first-degree relative using the offspring genotype is to multiply the regression coefficients, the effect sizes for the genetic variance, and their standard errors by 2 to correct for 50% attenuation due to 50% genetic relatedness. So in this paper, we developed and validated a multivariate model using genomic SEM that directly estimates the appropriate correction empirically from the GWAS and direct GWAS summary statistics in order to produce unbiased estimates of SNP heritability and also of individual SNP effects. So our multivariate model, as represented in this path diagram, integrates data from 3 sources. So we have direct GWAS, maternal GWAS, and paternal GWAS. So in this model, the total genetic propensity toward Alzheimer's disease risk is represented as a latent factor, this F factor, and that is specified to affect the direct GWAS phenotype, Y direct, and the 2 GWAS phenotypes according to this system of regression equations, where the lambda, the vector of lambda coefficients are attenuation coefficients relating the F factor to the measured phenotypes, And the U terms represent residual genetic propensities toward each of the measured phenotypes that are independent of the genetic factor F and are uncorrelated with each other and independent. So we impose a minimal identification constraint that the attenuation coefficient for the direct GWAS is 1, such that this latent factor takes on the scale of the direct GWAS, and the variance of the latent factor can be interpreted as an unbiased estimate of the SNP-based heritability. Derived meta-analytic summary statistics for this factor. We accommodate individual variants in the model to estimate the effect on this broad factor of genetic risk for Alzheimer's disease. So using simulations,
I guess just a quick question.
Yes.
Do you have to have separate maternal and paternal GWAS here?
Yes, exactly.
So
yes.
Otherwise the model wouldn't fit in that case?
I mean, you can definitely fit it just using either maternal or paternal GWAS.
Also, so you're saying if you have two nodes downstream to the shared factor, you could still fit a model. That's okay.
It wouldn't be identified. So ideally, at least 3 indicators.
Okay. Yeah.
For a factor model. Yeah, unless you have like an instrumental variable like a SNP to gain some degrees of freedom, but yeah.
Okay.
And of course, this can accommodate GWAS data from second degree relatives and so on. So using simulations, we compared the performance of this novel method that we developed with conventional approaches to recover SNP-based heritabilities and individual SNP effects across different conditions, mostly varying the assumptions of the standard GWAS model that the attenuation coefficients are 0.5 to represent this 50% attenuation expected for first degree relatives. And we found that our model exhibited consistently unbiased performance across conditions and outperformed conventional approaches in most conditions. So we then went to apply our model to empirical data. Using summary statistics from direct case control GWAS of Alzheimer's disease in the International Genetics of Alzheimer's Disease Project and maternal and GWAS of maternal and paternal history of disease from UK Biobank. So we first estimated genetic correlations among the indicators using the LD score regression. And we found that the correlations were not different from 1, indicating that they were not genetically distinct traits and supporting the usefulness of this GWAS types combined with case control G. So in the model, the SNP-based heritability in the liability scale, assuming a population prevalence of 5%, was over double the one reported in the largest GWAS to date, assuming the same population prevalence. This is excluding the Alpway region. Okay, so what we found that was interesting is that the unstandardized loading for paternal history of disease reflected approximately 27% attenuation from the expectations under the simple GWAS model. So we would expect to, it should be 0.5, but it's 0.37. So this means that there's further attenuation. This could happen when a set of participants confuse their current disease status, or it could also be due to differences in reliability from parental history of disease, which is based on self-reports compared to carefully screened participants for clinical Alzheimer's disease. And the residual variances represented in these U terms were very close to 0, indicating that the GWAS and GWAS were not tapping genetically distinct phenotypes to an appreciable extent. So,
your explanation there of the attenuation, the difference between maternal and paternal, is your view that you think that they could be the same, the 0.46 and the 3.6, and so that you don't need to have a paternal specific story of why the differential is bigger in the paternal, or do you think they're different, which is to say 0.46 and 0.36 are different, and the stories you're describing would need to have like differential reporting error when you report your dad's status than when you report your mom's status? Is this like, tell me what your thinking is here.
Yeah, so based on the genetic correlations that were not different from one, but the standard errors were a bit large. So
Now you're saying over here.
yeah, over there, yeah, and in the
You're saying that the
itself,
0.84 is not different than one from us because you can't rule out with big standard errors, but like 0.8 is not numerically that close to one, maybe.
Yeah.
So what's your view there?
Yeah, exactly. So I think it's a power issue because the number of maternal and paternal cases, the effective sample size was not very large. So the standard errors were quite large, and it's not easy to tell. We don't find evidence to say that they're genetically distinct traits, but it could be that they're in actuality different, genetically different traits. And in the models, I would have to take a look at the confidence intervals to actually get a better sense whether this 0.46 and this 0.37 are actually different. But yeah, definitely there can be systematic sources of bias in the attenuation coefficients that are different from maternal and paternal proxies. So, and that I think that's an interesting question that could be definitely assessed by incorporating other traits within the genomic SEM model to see how they relate with the residual variances and with the latent factor itself.
So was the motivation thinking like if you would find that the maternal like if the lower numbers like the residual, if that would have been for maternal, say, you know, larger, would you have thought something like mitochondrial DNA, or I'm what's the motivation?
So yeah, that's a good point. So if the residual was larger and significantly different from zero for maternal, but not for paternal, that would indicate that there's some uniqueness in the genetic architecture for maternal history of disease. But going from there to biological explanation, that's a long way to go, because it could be just different sources of bias. The way people report the phenotype of their mother could be more precise for some reason. Could be associated with I don't know, educational attainment or cognitive function. So yeah, there's definitely, but in this case, we didn't find evidence for significant residual.
Yeah.
Exactly. So yeah, overall, this indicates that integrating GWAX data with case control data it's a relatively valid approach. This. Okay, so we next assess the effect of assumptions regarding population prevalence rates on our liability scales and heritability estimate for Alzheimer's disease, and we varied the assumed population prevalence rate. Because in Alzheimer's disease, its lifelong prevalence rate goes from 1% in late adulthood to over 30% for people. I mean, from people at 60 years old, we find like a 1% population prevalence rate for Alzheimer's disease, but it goes up to 30% with age over 90. So we and the assumed population prevalence rate has an impact on the estimates of liability scale heritability. So we varied the assumed population prevalence rates, and we found that heritability estimates of Alzheimer's disease increase as the population prevalence rate increases.
What's the estimate based on the IGAP?
The prevalence rate?
No, just the heritability. So where would that be on the line?
No, this is based on our multivariate model.
Yeah, so I'm wondering, so if you just use case control GWAS, what's the heritability estimate?
We didn't test that across different population prevalence rates, but we did for assuming the standard 2.5, I think.
I guess my question was just, is it close to the eventual multivariate?
Yes.
Okay.
Yeah, they're very close.
Okay.
Yeah, so there's no such a big gap like it was between the reported meta-analysis combining GWAS and GWAS data with standard approaches
Right.
that reported 2.5%, and the case control heritability was more in the line of 9%, between 7 and 9%. So we were close to the estimates for case control data. So again, these two squares here, the yellow one is the estimate of heritability from our multivariate model, assuming a population prevalence rate of 5%. And the purple one comes from the largest GWAS today, assuming the same population prevalence rate. You see that it's more than double. And this dramatically downward bias was emerging from this integration of direct GWAS and proxy GWAS. Okay, so what we did next was to investigate local heritability. So how heritability was distributed across the entirety of the autosome. So we inferred common variant polygenicity from local SNP heritability analysis using approximately 1,700 independent LD blocks. What we found is that heritability or genetic risk for Alzheimer's disease was homogeneously distributed across the entirety of the autosome with a sharp discontinuity at the apoid locus, which we found that explained 10% of the total SNP. And the other GWAS hits, the genetic risk loci identified by GWAS, explained another 9%. So this leaves out approximately 80% of SNP heritability unexplained by apoE or the genetic signal captured by GWAS, which suggests that the genetic architecture of Alzheimer's disease represents a strong effect of apoE, of course, but it's superimposed on a highly polygenic and diffuse fact. Okay, so now I'm going to present some preliminary results on my ongoing work on dissecting the multivariate genetic architecture of health and aging with a special focus on chronic conditions.
I have a quick question.
Yeah.
For the previous paper. So essentially, the previous paper is just do a meta-analysis, but using a more flexible genomic SEM model. So have you compared your results with you simply using let's say using metal to do a meta-analysis?
Doing what approach?
Using metal, just do a very simple, like
Yeah.
an inverse variance-based meta-analysis?
Yes, actually, in our simulations, we compared the performance for method represented by the purple distribution. So the red dashed line is the true, like the parameter in the population that we simulated data from, and the purple distribution is the heritability estimates from our multivariate model across 100 iterations, and the green and the yellow one are the standard conventional approaches. With the green one is the uncorrected approach, is the sample size weighted meta-analysis, and the yellow is the conventional approach with the standard correction of multiplying by 2 the effect sizes and the standard. So, of course, in conditions where the simple GWAS model assumptions are correct, like attenuation coefficients are exactly 0.5, but we don't find that empirically. But just assuming that that's correct in the population, our method and the conventional approach with standard correction, they will provide unbiased estimates. But in more realistic scenario, our model still recovers unbiased estimates.
So the yellow one is always underestimated.
Yes, exactly.
Can you comment on the eventual effective sample size you get after doing the meta-analysis?
The effective sample size for our multivariate model.
Yes.
I can't remember exactly, but I would say it was approximately 90,000, the effective sample size.
But I guess just relatively, if you compare that with say fixed effect meta-analysis, I know it comes with bias, but is there any like attenuation in the sample size you get after using this multivariate model technique?
Yeah, we do, because for example, so the calculation of the effective sample size for for latent factors, common genetic factors, it's a bit tricky, and in this case, it's a bit more complex because you need to correct the fact that the family history of disease GWAS data is already attenuated for first degree relatives. So we use the attenuation coefficients empirically found in our multivariate model to correct the original sample sizes for maternal and paternal GWAS. So basically, we multiply the effective sample size for maternal history of disease, multiply it by 0.36, and we sum that to 0.46, the attenuation coefficient for maternal, times the maternal sample original sample size, to derive a composite effective sample size.
I see. So is it fair to say that suppose the attenuation coefficients are exactly 0.5, which is, I guess, what other traditional meta-analysis would assume? In that case, your sample size in the final output would identical to what you get from traditional approach.
Yes, exactly. Yeah, it is actually possible to perform a manual correction to the sample sizes to put the summary statistics into L-score regression. The proxy GWAS phenotypes. Okay. But that's typically not done.
Okay.
Okay, so again, I'm gonna provide a brief overview of my ongoing work on genomic architecture of human health and aging. I recently submitted this project as an R1 grant, and I'm going to provide some preliminary results that I think are very exciting. So the overarching framework is based on the concept of healthy aging as a life course process of developing and maintaining functional ability that enables well-being age. Functional ability comprises the health-related attributes that enable people to be and to do what they have reason to value. And it is made up of an individual's intrinsic capacities, which is a composite of all physical and mental capacities, and the relevant environmental factors and the interactions between the individual and this factor. So through the lifespan, there are periods with relatively high and stable intrinsic capacities, but with advancing age, there are declines in capacity, which ultimately result in a significant loss of physical and mental capacities. And of course, the environment can exacerbate or mitigate these declines in intrinsic capacity. So yeah, so aging is a multidimensional process highlighting both genetic and environmental factors and a complex interplay between these factors. It is associated with diminishing capacities across multiple physical systems and is the main risk factor for chronic disease development and accumulation. It's important to note here that there's a remarkable variability in the way people age at both phenotypic and biological levels. So at the biological level, aging is associated with a gradual accumulation of molecular and cellular damage over time, resulting in a series of changes that have been summarized in known as hallmarks of aging. So this figure here covers some of the most relevant hallmarks of aging. So this include DNA damage as a result of gene expression and copying and DNA damage as a result of copying and gene expression errors. Proteins become less functional. There's an inefficient cell communication, imbalanced metabolism, telomere sorting, and so on. So eventually, these broad changes manifest at a clinical level as chronic conditions. In that regard, that's why advancing age is associated with an increased risk for multiple chronic conditions across diverse organ systems. So multimorbidity, which is the coexistence of two or more chronic conditions within an individual, is highly prevalent among older people. So this plot here shows the percentage of patients with different number of chronic conditions across age groups. We can see that more than 50% of people over 65 years present 2 or more long-term conditions, resulting in complex multimorbidity patterns. Multimorbidity has been associated with the suprafunctional decline and worse quality of life, and of course with elevated healthcare costs. But how does biological aging enable chronic disease development? So the neuroscience aims at linking aging to chronic disease development and suggests that aging increases multimorbidity risk through shared biological mechanisms of physiological decline across multiple organ systems, such that better understanding of the biology of aging could be helpful to identify molecular targets, to develop drugs and integrative interventions aiming at slowing, even preventing morbidity and improving health and well-being in the old age. So in that regard, genomics provides a tremendous opportunity for clarifying general or shared biological mechanisms underlying system aging. A formal investigation of how genetic variation, and more specifically how genetic pleiotropy, increases the risk for multiple chronic conditions across diverse organ systems can be useful to identify genetic variants roughly relevant for health and aging, and to potentially to provide critical insights into general aging processes. As I showed before, recent GWAS of medical traits highlight substantial genetic sharing among diverse chronic diseases of aging, and these are exciting results, but again, they are very limited in the sense that they are focused on single diseases and they do not capture the bigger picture. So this is what one of the motivations to conduct a formal investigation of the joint genetic architecture, the most common and prevalent and burdensome chronic conditions in the old age. So so far, we have explored and identified clusters of age-related diseases with varying degrees of genetic overlap using GWAS summary statistics for these 24 chronic conditions from some of the most prevalent and burdensome disease categories, including diseases of the circulatory system, respiratory, and musculoskeletal diseases. Of course, I plan to expand the number of domains. I have already worked a little with metabolic diseases, cancer, and neurodegenerative disorders, but these are the preliminary findings that I would like to share with you today. So as highlighted here in the genetic correlation matrix among these 24 diseases, we found substantial genetic overlap among circulatory, musculoskeletal, and respiratory diseases. The best fitting genomic SEM model was this genetic bifactor model, suggesting the presence of a general disease liability factor, accounting for the shared genetic variance among these 24 diseases of aging that are manifested across different tissues and organ systems. So this factor represents, we think, it could represent a fundamental dimension of genetic risk sharing across age-related diseases that could be potentially revealing a common aging process, multi-system physiological dysregulation. But we additionally found these 6 specific factors of transdiagnostic risk for more specific clusters. So the first is a transdiagnostic factor for coronary diseases, grouping conditions affecting the blood vessels that supply blood and oxygen to the heart muscle, like angina, myocardial. The next factor is an arrhythmias factor, which groups conditions that affect the electrical signal that regulate heartbeat, like atrial fibrillation or other arrhythmias. There are also specific factors for coagulation diseases, diseases of the connective tissue, back-related diseases, and respiratory diseases. So the next step is to perform multivariate GWAS discovery on these broad factors of genetic risk sharing. So basically incorporating individual variants in the model to identify regions in the genome with broad effects on multiple chronic conditions. So we have already done that using the respiratory factor, which because it was one of the more simple factor solution, it only has 3 indicators. So we conducted multivariate GWAS on this factor using 7 million common variants. So in our model, it's very similar to the Alzheimer's disease model. The general risk toward respiratory disease is represented as the latent factor RSP, which directly affects the genetic component of chronic obstructive pulmonary disease, COPD, asthma, and upper respiratory diseases. The factor loadings, these lambda coefficients relate the factor to each of the individual diseases. Few terms represent residual genetic risk that is specific to each particular disease. Our model, of course, accommodates individual variants, which allows to identify these regions in the genome with broad effects on respiratory function. So this is the Miami plot for the multivariate GWAS on this broad respiratory factor. We identified 94 risk loci. 20 of these risk loci were new discoveries unique to our multivariate approach, meaning that they were not previously identified in the univariate GWAS of the individual diseases. And these are represented as the red triangles. Okay, so the blue triangles represent the other 74 genomic that were common with the univariate GWAS. But importantly, it's even more important the fact that these 74 genomic regions that are associated with broad respiratory function and that were previously reported to be associated with some of the individual diseases are not just relevant for a particular disease, but rather they're broadly relevant. So we also identified 27 QSNP hits, indicating genomic regions presenting more heterogeneous patterns of associations with the individual diseases. So just an example, the nearest gene to genomic risk locus 6, one of these new multivariate discoveries, is near the cadmium-3 gene, which is a tumor suppressor gene implicated in cellular senescence, which is one of the hallmarks of the gene. Of course, the meta-analytic GWAS summary statistics derived from these multivariate meta-analysis on clusters of multimorbidity and diverse transdiagnostic factors for diverse chronic diseases can be used to derive polygenic risk scores for different multimorbidity clusters, representing as a weighted additive effect of the allele, the risk alleles, growth factors, genetic risk sharing. This could be useful for risk stratification, of course, to address some developmental questions, see how this, for example, how these polygenic scores for multimorbidity patterns are associated with rates of change in accumulation of chronic diseases. Okay, so another very interesting application of genomic SEM allows the identification of risk and protective factors to transdiagnostic disease liability. The multiple indicator, multiple cause approach in genomic SEM, as I mentioned, allows testing the generality versus specificity of associations between correlates or of transdiagnostic risk factors of chronic diseases. So this is the path diagram for the mimic model, for this genetic mimic model. For three external correlates. So these X variables here represent the genetic component of three external correlates. This could be physical activity, educational attainment, or smoking behaviors. And a single common genetic factor underlying a number of diseases as represented by these factors. So this model tests whether the collateral phenotypes or the external phenotypes are associated with shared genetic liability across diseases as represented by the solid paths from the external correlates to the F factor, or whether they're more specifically associated with individual diseases as represented by the dashed lines. Of course, this model can be extended to allow for additional phenotypes and multiple factors. So in conclusion, just some take-home message. So first, multivariate genomic methods as the ones that I described provide tremendous opportunity for clarifying shared biological pathways underlying multi-system aging. They allow to address some research questions into multidimensional phenomenon from a different angle compared to classic approaches. A formal investigation of genetic risk sharing among chronic diseases of aging is promising in identifying targets for integrative interventions aiming at improving health and well-being. And finally, as shown by these mimic models, leveraging the advantages of large-scale GWAS, of behavioral, lifestyle, and medical phenotypes, we can test behavioral, psychosocial, and medical pathways of risk and resilience to it. And thank you all. Yeah, that was all. Thank you.
Yeah, thanks for the presentation. I was at your job talk yesterday, so this is the second time I've been able to listen to you talk about it. And if I've missed something, it's my fault, not yours. But I'm pretty sure I didn't hear you talk much about really in your R01 proposal and in well, for the APOE study that was UK Biobank, but for this study, I didn't hear you talk much about the population that you're interested in. So, for instance, all of the GWAS summary statistics that you were using, are they mostly based on European ancestry individuals? And for the PGS study, right, the PGS application, what was the target sample? Was it also European? And then if so, or if not, like, what is your thinking around the portability of everything that you're doing, the identification of these really important genes that are relevant for general aging issues and the extent to which they're actually identifying multi-ancestry? Can you speak to that?
Yeah, thank you. That's a very relevant question. And yeah, so far we've employed GWAS summary statistics from European ancestry because they're better powered, and for a starting point, it was a good point to start because it was easier to get it. But we definitely want to not just to replicate, but explore the genetic architecture in different ancestries. So there are other resources like the global meta-analysis, the Biobank initiative. So we plan to explore this factor models in different populations. And that would be the most direct way to provide evidence for shared genetic architecture across different populations. But we're planning also to test the predictive ability of PGS derived from these models in different ancestries in longitudinal studies with diverse ethnic backgrounds.
Okay, I'm going to follow up with that on that question because it's you mentioned this. It's not just genetics, right? Mm-hmm. So you even said in one of your bullet points that it's really important to also account for gene environment interplay, mm-hmm. especially as it relates to aging.
Exactly.
And so, whether this related to ancestry or not, how do you conceptualize environment in the work that you're doing? Is it sort of like a separable component from genetics, or have you thought about environments in ways that actually influence genetic discovery? I'd like to hear your thoughts about that too.
Yeah, that's a very interesting point as well. So, as genomic SM is a very general framework, ideally, we could use GWAS summary statistics from within family designs and compare the fit of the models and the parameter estimates from covariance matrices that include this indirect effects from covariance matrices that actually discount this effects on the covariance structure. So as long as there are GWAS sumstats from other designs like within sibling, that could be a way to test whether a substantial confounding in the sense of indirect genetic effects. But from there to actually understand what type of effects and biases are underlying the covariance structure, it's a complicated question to address, but yeah, I would like to.
Okay, yeah, we'll talk more later. I like to ask one more question if you
Yeah.
don't mind, because this has been on my mind for a while, or at least 24 hours. You know, we're very familiar with GSEM here. We know the group that you work with quite well, I think. And I think one of the, you might have received this question yesterday. I'd like to ask maybe a very similar question, but you can answer it in a different way. Like, what is the clinical utility of detecting a D factor or P factor? So I'm a psychologist. We know the P factor exists, both in phenotypic studies and in GSEM, but it's clinically irrelevant. Like there's no specificity for a clinician, right, to know that you have a general liability for psychiatric risk. It's etiologically interesting, but it doesn't provide much clinical value. So I guess I'm wondering, given your discovery in, I think it was, yeah, your preliminary results for your R1, the detection of this, general liability factor for aging. What is the clinical value of that? What is it supposed to represent? Is there anything like real about it? So just when Andrew was asked the question of what is a P factor, I think he said something, oh, it could represent neuroticism, right? Or some other psychiatric trait that we might have to measure separately. But no, I'm curious to hear what your thoughts about what that is and what the clinical value of that is.
Okay, thank you again for your question. So I guess for the D factor, for this general disease liability factor, it would be harder to derive clinical utility other than maybe using these GWAS results to compute polygenic scores and somehow as a global propensity towards a better health status. But for the more specific clusters of transdiagnostic risk for circulatory diseases or musculoskeletal, I think it could be there are interventions that could be definitely targeting specific groups of diseases like physical activity or strength training could prevent like some musculoskeletal diseases or the loss of muscle mass. Just I'm coming up with these ideas now, but I guess that for the specific clusters of transdiagnostic risk, we have the application of stratifying individuals based on their genetic risk. And another promising area in development is precision medicine. So how different genetic profiles in terms of genetic propensity toward different multimorbidity clusters are associated with how people respond to different treatments, both drugs but also like other types of interventions. So yeah, and for the D factor, I guess it is promising. It could be promising in identifying molecular targets to develop drugs that have cross-trait effects. But yeah, that's all speculation. We need to conduct further analysis to see, because this D factor might be just associated with educational attainment,
right?
Like lifestyle behaviors, and it's not really something purely like a senescent or aging process, clear biological implications.
Okay, thanks.
Quick question is about in general about the genomic SEM model. So for some of your model, it looks very complicated. So I think when there are many factors in your model, theoretically you could have maybe a millions of models to choose, and then you choose one. You think maybe based on some theory, you think this is a reasonable model, and you fit data and data support this model. But my question is, how robust is the finding? Maybe you could find a totally different model, and then the data could also support that model with a totally different interpretation. So how robust is the model? How do you verify the finding is indeed a correct finding?
Thank you. That's a very good question. Structural equation modeling, both at phenotypic and genetic level, is a confirmatory approach. So ideally, it should be based on theory, as you said, like you have a clear idea or strong hypothesis of the genetic architecture or how these factors relate to the measured phenotypes, then you should definitely go to test your theoretical model. But there are scenarios where there's no much theory, right, on the underlying mechanism. So in that case, there are some exploratory approaches. So for instance, here, what we did is, of course, we could start fitting models based on organ systems or disease categories. Like, let's see if the circulatory disease category is something at the genetic level. But the approach that we followed here consists in splitting the GWAS summary statistic file into odd number autosomes and even number autosomes. So we conduct exploratory analysis in odd number autosomes using principal component analysis and exploratory factor analysis to kind of identify clusters of traits with a shared genetic basis. And then we use that information, purely exploratory, to confirm the models in even number of autosomes. And that's a pipeline when you don't have a clear idea of what's going on. But yeah, this models are sometimes very unstable. Like if you include an additional trait, the fit goes wrong, residual correlations appear, there are convergence issues. So what you should never do, I think, is to capitalize on residual correlations to improve model fit, like start tweaking your model just to get a good fit, because that might not replicate in an external sample. And of course, trying to replicate the models in external samples. So this, I mean, the biggest resources of this subsets of conditions, of course, UK Biobank, but also the FinGen Biobank. So a good way to provide evidence for replicability be to fit the same model in these two separate data sets. But again, yeah, one very important thing is to conduct this analysis in ethnic backgrounds, because we're talking about general aging processes. If it's something that is general, it should be found in different across ancestries.
Yeah. Two questions about the method you presented. The first one is probably well understood in psychological sciences, actually, and it's related to Yuchang's question as well. I'm wondering if you want to find the shared factor across many different phenotypes. Suppose constraints in data is not a concern. I suppose you could just do PCA on everything and do a GWAS on the first principal components. I just wonder, how would you compare that approach with that single shared factor based on a structural equation model and then GWAS on that? And the second question is, initially when I presented this multivariate meta-analysis approach, I didn't have the intuition why it would work, because to me, it sounds like you're doing some kind of average of case control GWAS and maternal paternal GWAS. So then why does it favor the case control GWAS so much in your results? But then I realized you have this fixed edge of one pointing towards the case control GWAS. So then it makes me wonder what's stopping us from doing this all the time for all GWAS. For example, if I want to do a GWAS on, I don't know, depression turns out to be underpowered. What's stopping me from doing this by combining 200 published GWAS, but then just fix the edge to be one on depression? but then everything else can be always incorporated into the model. So would that lead to any problems?
That's very interesting. Actually, it's a very good idea, I think. I mean, of course, so fixing to one, the factor loading relating the factor to one indicator that makes that indicator the canonical indicator. So that the factor takes on the scale of that GWAS, and it's basically representing the same trait or construct. Yeah. So if you incorporate like genetically correlated trait, that's very similar to what MTAG does. But in this case, in genomic SEM, compared to MTAG, we have Q-SNP statistics that safeguard against false discoveries to distinguish genetic variants that are broadly relevant for your factor versus genetic variants that are more specific with a trait. Because if you incorporate like lots of traits, you want to be able to distinguish if the SNP is actually operating through the latent factor versus.
But that's what the Q statistics.
Yeah, exactly. So just saying that, yes, what you say, it could be definitely something feasible, but not sure if at a substantive level. I mean, at first it sounds like reasonable, but I would have to think and discuss this more calmly. Yeah, sounds like it could be definitely possible. And the other thing, the distinction between the genetic factors and the PCAs. Yeah. So PCAs, it's about what this two approach assume. So PCA is just a linear combination of the original variables to maximize the variance, right? But they don't assume an underlying factor or mechanism causing the observed covariance structure, which factor models do. So that's the main difference. And in terms of equations, PCAs, I think the PCA is in the left side of the equation, right? And in factor analysis, they're on the right side. So they're explanatory variables. So on the left side, we have the observed phenotypes. And on the right side, we have the factors and the lambda coefficients relating the factor to the phenotypes.
So is this more about factor analysis results being easier to interpret, or is this of empirical importance as well?
So I think that
for example, right, I've seen people doing GWAS on, say, risk tolerance, and one of the things they did in that paper was to use a variety of ways to define risk tolerance. I think they had smoking, they had alcoholism, number of things, right? And then they did the PCA and did a GWAS on that. At the time, I thought, it sounds like a straightforward, easy to understand approach, but maybe at the time GSAM was not even introduced. These days, GSAM become more popular. I just wonder, so if you actually do a GSAM on that type of data, would you actually get very different results? Do you expect to see different results, or empirically it will be kind of the same thing?
I do expect different results, because for PCAs, I think the polygenic signal could be contaminated and not reflect just the pure shared genetic variants.
Okay. And just elaborate a little bit on that, why it may be contaminated.
So, because the PCA might be capturing variation that is not shared among all traits included, but just like, for example, if the first two traits they share a lot of variance, so PCA might be mostly reflecting variables 1 and 2. Whereas factor analysis would be actually capturing the shared variance among all traits instead of capitalizing on like the strongest shared variance among all traits. Sure, that was.
I think I understand what you're saying, but it's just obviously this is eventually a numerical issue. But suppose two traits are very strongly correlated and drives the variance across all phenotypes. You would assume the loading to that first PC would be weaker from other traits. Just like in a single factor model, the factor loading to other phenotypes could be closer to 0. So I guess for example, in the single factor loading model, the edge towards half of the trait is very strong, but to the other half is much attenuated. Would you still call that the factor that's shared across all traits? Because you do have a lower weight pointing toward other traits. So I guess I don't understand how that's different from, for example, PC1 is more driven by half of the phenotypes, less so by the other half.
Yeah, I see your point. So I probably wasn't very clear about the thing about the shared variance in the two models. So I guess the clearest explanation and the most stark difference is in the assumption of an underlying unobserved factor causing the covariance structure versus just deriving a new variable, which is a linear combination. So that doesn't assume that there's any factor or latent factor underlying anything.
Mm-hmm.
So I think that's the biggest difference.
Okay.
I guess so. It's the assumptions, and mathematically, like factors are on the right side of the equation, whereas PCAs are just new variables on the left side.
Okay. Yeah. Thank you.
Well, your first study, I think, is an example when you had the response time was kind of an outlier among all the. So yeah, so is that relevant in this conversation that you could? Is that when you see something like that where your response time, as you'll see, or as you'll remind us, is an outlier in terms of its weight compared to all the other ones? Did that ever suggest response time's not the right? We should get rid of response time?
No, that's a good point, but again, as models should be based on theory. So even if it's so it's still part of cognition, right? So even if it's genetic variance,
I think this is an example where your theories,
yeah.
Where if you would have done factor analysis, the factor analysis might have suggested that it doesn't like load very well and get rid of it.
Yeah, exactly.
And where the theory says like I have to keep it. Is that right?
Yeah, exactly. Yeah. Yeah.
I'm interested in the overlap between kind of what you said in the first part and second part. So did you find any shared, I mean, for example, response times that maybe somehow could that perhaps go with more physiological aspects of the body? You could imagine that being maybe more related to exercise or physiological function and things like that, rather than a pure cognitive. Did you see any split of the cognitive function versus some of the other physiological things in the last part of the presentation? I didn't see if there was cognitive function thrown into all of the different.
Thank you for pointing out to this aspect, but yeah, we haven't integrated the cognitive function with chronic conditions of aging, but that's definitely a line of work that I would be interested in.
Yeah, I think that'd be fascinating because they're seeing more in midlife that these things are very, BMI and all of, being overweight, not, smoking, all these things are contributing to declines in cognitive function, but it also has to do with vascular disease and things like that. So I'm just curious whether some of these things are overlapping, because I think that is this big mystery in aging, or how these things are coming together to contribute to dementia versus a decline in other physiological systems.
Yeah, that's definitely a good point, and I would be very interested in integrating the genomics of cognition with the genomics of general health and chronic conditions, and also psychiatric health. It's like, I think there are lots of lines of work.
That's
to be accommodated in this, like
dementia.
The genetic architecture for major psychiatric disorders relate to more below the neck conditions at the genomic level. Be definitely interesting.
What's the genetic correlation between that G factor and EA?
It was in line of 0.4. 0.4.
I also just had a question of whether you've tested. I like this idea of kind of a polygenic score for aging almost, because it is, I'm an applied researcher. I'm always looking for tools that methodologies out there to apply to my research on aging. And that's been a frustrating component of research on gene environment interactions with aging, because it is this multi-system, issue and decline, and you can't just choose one polygenic score to try to model that. So I really appreciate what you're doing here. And I just wonder if you've actually looked in the health and retirement study, for example, to see if this polygenic score is more predictive of, declines or increases in the number of chronic diseases or things like that, as opposed to maybe some other polygenic scores that a person might use, or if you just tested it more in an empirical context.
That's it. in my list. So it's actually one of the specific aims of the R1.
Nice, okay.
The last aim after all the analysis on at the genome wide level and integrating PGS into longitudinal studies of aging and see how they will predict like health-related outcomes, functional decline.
Yeah. Yeah, and I could also see, applications. I mean, it's hard because there's not a lot of studies that have genetic data and epigenetic data, so it's hard to do these, but, with all the epigenetic clocks, there's so many different clocks, they're all capturing slightly different things to try to get like a shared genetic factor or to try to piece out what genetic components some clocks are capturing versus others. I think that could also be a really useful or interesting application.
Yeah, definitely.
Biomarkers of aging,
Yeah. Yeah, so for the mimic approach, yeah, I think I have already tested the association between some external correlates and these growth factors of genetic risk sharing. No at the disease of the general disease liability factor, but for the transdiagnostic risk factor for respiratory diseases, I have already tested the associations, and some of these external correlates are epigenetic clocks of biological aging. The genetic component of those clocks, because there's genetic variation contributing to epigenetic variation. Yeah. So there's some interesting associations, but also with BMI and other blood markers like inflammation markers, C-reactive protein, interleukin-6. So
Interesting.
yeah. So as long as there's well-powered GWAS for any trait of interest, it could be incorporated in these models without the need for raw genotype data, which is a promising thing.
We just need larger GWAS of
everything.
things like that. Yeah.
Two questions. I think the first one is a short one, which is in your big chronic model, where there's a factor at the bottom. Let's see. Yes. What's your view of how the general disease liability factor should be, at what level it should be correlated with like a lifespan GWAS, essentially?
That's a good point. So,
because you have parental lifespan in one of your
Yeah.
previous slides, which is not, it's not exactly what I'm asking, but it's on like how bad would this general disease liability factor be correlated with something like lifespan for you to say like something's wrong with my model?
That's a good point. I think I have some preliminary results at the individual diseases level with a parental lifespan, and they were all this, and also at the level of the transdiagnostic risk factors, and they all presented negative genetic correlations, significant and substantial. So yeah, that was one of the checks that I was doing. Like, okay, so things are going in the right direction.
Well, now that I see this, I was going to ask whether this model was in conflict with your respiratory model, but I think it's not, because your respiratory model here assumes it's its own separate thing.
Exactly.
That could be run separately. And this is, would you say that, I mean, this is a proposed model. Have you tested this to say, now maybe losing one of the slides, whether respiratory is on its own? And then therefore your next set of slides are testing a reasonable model? Have you confirmed that respiratory is on its own?
Yes, exactly. So yeah.
Or is
So
that, I guess that's to do.
I actually changed the pipeline because I've been updating the number of phenotypes to be as comprehensive as possible. So at the beginning, so it's very hard to integrate all these 24 diseases to run the multivariate GWAS directly in this model. So I had to separate parts of the model to conduct multivariate GWAS on within disease categories where there's substantial genetic overlap. And I'm deriving a new set of summary statistics for each of these specific factors of transdiagnostic risk.
And are you going to, so it sounds like I think you just answered this, but how much trouble are you in on the left-hand side here where the three factors are overlapping in this way? Are you going to be able to run this?
And
so if you cut the model in half and look at the left-hand side where there's a lot among the factors, is that going to, have you already decided that that's going to be a very difficult model, or are you already?
Yeah, it's going to be computationally intensive. And probably find some issues with convergence and stuff. But yeah.
Because I think Lauren's point that you agreed with was that you're in the business of expanding further that cognitive part, and you have other things. It's like a psychological part or something. Seems like it's going to get more and more difficult.
Exactly. So my solution for that but I think it could solve this problem is to first reduce dimensionality within disease categories, like running multivariate GWAS on respiratory factor, like I've done. So you reduce from 3 indicators to 1 set of meta-analytic substats. So I'm going to do the same for all these transactions and then put them all together again and see if this D factor still holds, and it becomes like more feasible in terms of computation. But yeah, that's one of the most complex parts of this part of the project. Like, am I going to do it separately, altogether? And what happens when I expand the number of disease categories? Yeah. Yeah, for cancer, for example, I haven't found any substantial genetic overlap among different types of cancer. Talking about this deal. Same for neurological disorders. No substantial genetic overlap between Alzheimer's, Parkinson's disease, and the other traits.
Have you tried throwing like smoking GWAS in there, or some of these like really, low-hanging health behaviors that we know contribute to accelerated aging and earlier mortality, just to see if there's any shared genetic architecture, like excessive drinking or alcohol use disorder or smoking or.
Yeah. I actually, let me see if I have sure. No, I don't have the a hidden slide, but yeah, for yeah, so at the level of the this transdiagnostic factor for respiratory diseases, I have assessed the relationship between this factor and 37 external correlates, including smoking behavior, alcohol consumption, physical exercise, educational attainment, BMI, and many others. And one of the external correlates that presented broad effects on 3 diseases that basically that was operating through the general factor was smoking. It was expected, but it's cool to see that it's working.
Have you tried to put the GWAS of smoking into the factor model, or
as an external correlate, like predicting?
Right. But have you tried to throw it in there?
Yeah.
And it doesn't swap, like take everything away. It's still
it's still
own. Does it load onto its own factor, or does it load onto the respiratory factor if you put the smoking GWAS in there?
So let me find the slide. So I put smoking like one of these X circles here, like as an external correlate of the respiratory factor. That would be the F factor, but it's the respiratory.
Oh, I see. Okay.
So it's like you estimate like a beta, an effect of the external correlate on the general factor.
I thought Lauren was asking if you said that smoking was one of the Y's on the right.
Yeah.
Right?
Yeah.
So put it
on.
Yeah.
Did that?
No.
You haven't tried that, because I'm just curious if that variation in smoking genetic architecture might, I don't know, swamp some of this other variation or switch up. I don't know if it would change the loadings, or if it would load onto the respiratory factor, or
Yeah, that could be definitely possible, but theoretically, like we want to have like a pure medical factor, like a capturing genetic risk sharing among chronic conditions, not smoking behavior, like it's considered like an external correlate, like a risk factor rather than a chronic disease itself. So it would be theoretically more difficult to interpret the factor if you like.
We're about at time. Thanks so much, Javier.
Thank you so much.